Good afternoon, all of you. Today we are going to continue with the part sexual reproduction in flowering plants, and today our topic of discussion is megasporogenesis. In our previous class we have studied about microsporogenesis. We have seen that how the pollen grains they are forming and what are the different steps behind that. Today we are going to study about megasporangium and how the megasporogenesis is taking place. The development of ovule or embryo sac that comes under the study of the female gametophyte. Yes, we are going to study about the developmental procedures which are taking place in case of female gametophyte and how those are taking place in a stepwise manner. At the very beginning, we must know the structure of a pistil. Before starting with the structure of a pistil, I would like to show you a freshly collected flower and to locate the different positions in that flower. This is the flower of Liliaceae family. We are going to have practicals on that. So as you all can see here, this flower, a brightly colored flower and also a large flower, you can understand. There are certain places where the anther lobes are present. These anther lobes, they are having pollen grains in them. If you observe minutely, you can understand some yellow colored materials, those are coming out of that. That is related to the process of microsporogenesis. But today we are going to study about megasporogenesis. So right at the base, if you observe, here you can understand this base is bit bulbous. Actually, this is the ovary part where the ovules are present. And these ovules, they are carrying the embryo sac inside them. So if we see the structure of this flower, this is a complete flower and we know that both androecium, these are the androecium which you can see here, the androecium and the gynoecium both are present here. Right at the center of this flower, the stigma is present. This is the topmost position of the stigma. A very long style we can see here which is extending inside the flower and it is coming here up to this portion in the ovary part. So here in this flower, this is a complete flower. We are going to study about megasporangium. So it is just an idea that which part of a plant or a flower we are going to study about. So we are going to study about the ovary. Inside the ovary, ovules are present. Inside the ovules, how the gametophytes are present there. That is our topic of discussion. So let us begin with the part here. It may consist of one or a number of carpels and one carpel is having three parts. What are the three parts? The stigma, the style and the ovary. Generally, this basic concept we are having that one pistil comprises of three parts. Stigma, style and ovary. The topmost part which is flat and generally they are receptive in nature. They are receiving the pollen grains. So there we find that they come up. The stigma, the pollen grains, they get dust over. So the flat part at the top, that is the stigma, there's a long slender style which is extending up to the ovary and inside the ovary we find that ovules are arranged and they are having certain patterns of arrangement. So there we find that ovary, it is the swollen basal part of the carpel. Inside the ovary there are locules which contain the ovules. Each ovule encloses an embryo sac. Inside the ovule, we find that embryo sac are present. Now, formation of ovule or megasporangium. We say the ovule as megasporangium because inside the megasporangium, the megaspore or the embryo sac inside the embryo sac, the egg cell is present, which is very important for the process of sexual reproduction, which is carrying the n number of chromosome or haploid cells. So let us begin with the part. An ovule is called as a microsporangium developed as a tiny knob in the ovary. It is having a short stalk known as funicle and the body of the ovule fuses with the funicle in a region called hilum. When you are practicing the diagram of one ovule and megasporangium structures, there it's very important to locate the funicle part and the hilum part. These are very important for labeling your diagrams. The multilayered body of the ovule is called nucellus and it is enclosed into two protective layers called integument. So the major part of the body is composed of nucellus and there are two walls, you can say two jacket-like structures which are present there and those are the integuments. Integuments are having a small portion which is not a continuous part. Rather, there's a small area, except that area the protective wall is not present. A small pore is present at one of the end, and this area is known as micropyle or micropylar region. So far what we have seen, the structure of the ovule is known as megasporangium. In case of megasporangium, there are certain points which we need to go through, like the funicle with which it is attached with the ovary part, ovary wall, and the small knot-like structure is known as hilum. The main part of the megasporangium, that is the nucellus, and it is protected by two outer covering called integument. Integuments are not thorough, somewhere they are not having the integumental protective layer, and that particular portion which is forming a pore, that is called as micropyle or micropylar end. Now, within the nucellus is present one megaspore mother cell or an embryo sac mother cell. We are saying it as mother cell because this cell will undergo with the process of meiosis. The other end of the ovule, opposite to the micropylar end where funicle joins with the nucellus and integument, is called chalaza or chalazal end. It represents basal part of the ovule. So, if we see the parts of a mature ovule, what we find? Integument, 1 to 2 protective layers of ovule. Micropyle, a small pore at one end of the ovule through which pollen tube enters. This micropylar region is very important because that is the particular area where integuments are not present, the protective walls are not present. So at the top, on the stigma, when the pollen grains have fallen, and if those are compatible type, they will be obviously flowing down where they are going to enter inside the embryo sac, and through the micropylar region they are going to enter. Nucellus is a multi-layered main body of the ovule, and embryo sac present within the nucellus, which is containing 8 cells. 3 antipodal cells opposite to micropylar end, 1 egg cell, 2 synergids, and 2 polar nuclei in the center. So, we are going to study about anatropous ovule, and you must practice the diagram of longitudinal section of an anatropous ovule. How the integuments are present there, how the nucellus is forming, and how the cells present inside the embryo sac. That is very important part under the study of megasporogenesis. Development of megaspore mother cell. So, let us study with the different steps of megasporogenesis now. Megaspore mother cell is a large cell containing dense cytoplasm and prominent nuclei. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. In case of ovum, we know the egg cell particularly, we know that they are also undergoing with the process of meiosis. So this megaspore mother cell, they are undergoing with the process of meiosis, and they are forming 4 haploid cells. Out of these 4 haploid cells, only 1 cell is viable, and what is happening to the other cells, let us go through. They are actually degenerative type. Development of megaspore. A megaspore mother cell is a large diploid 2N, that is having both the sets of chromosome, 2N cell. It undergoes meiotic division and give rise to haploid cells or megaspores. In a majority of flowering plants, only one of the megaspore is functional and the rest 3 degenerates. Generally, we find after the process of meiosis, 4 cells are produced and all the 4 are haploid type. Now, all of them, they are not going to get generated. Only 1 is functional and the other 3, they are degenerative type. They are non-functional type. The functional megaspore grows, its nucleus enlarges, undergoes mitotic division and produces 8 haploid nuclei. The structure formed is termed as embryo sac or megagametophyte. So inside the embryo sac, how the cells are present? Actually, they are composed of only 1 haploid cell. This 1 haploid cell is undergoing with the process of mitosis. So when they are undergoing with the process of mitosis, this is very important that same number of chromosomes will be retained there. So the method of embryo sac formation from single megaspore is known as monosporic development. The kind of development described here, we are going to study about the processes, how in a sequential basis the cells are undergoing with the step-by-step division, ultimately giving rise to the embryo sac. The nucleus of the functional megaspore divide mitotically and form two nuclei, and they move to the opposite poles. So at the very beginning, we find that mitosis process is undergoing, and after the process of mitosis, from one nucleus, now two nucleus have formed. These two nucleus, they again undergo with the process of mitosis. So on the one side 2, in the next end 2. That means total 4 nucleus are present. All these 4 nucleus, they are again undergoing with the process of meiosis, totally forming 8 nucleus. All these 8 nucleus, they are getting oriented in a proper manner. So how they are getting oriented? We find that 3 are present at the chalazal end, and those are known as antipodal cells. 3 cells are present towards the micropylar end, and there we find one egg cell, that is functional egg, and other 2 supportive cells are there, which are known as synergids. Right at the center, there are 2 polar nuclei. So what we find at the top, there are 3 cells, antipodal cells. Towards the down also, we find there are 3 cells, 2 synergid and 1 egg cell. Right at the center, there are 2 polar nuclei. Actually, there are 7 cells but 8 nucleus. Try to understand. 3 cells at the top, 3 cells at the down, 6. In between, there's the central cell comprising of 2 nuclei. So here this is a binucleated structure. In some plants, you may find that there's a separating wall in between, but generally we find there are 7 cells but 8 nucleus. Sometimes this comes as a question that describe this point. There are 7 cells and 8 nucleus, and how they are present, in what basis they are present, that you need to go through. So once again, let us see how the process of division is taking place. Now in this diagram, let us see the process of megasporogenesis. The first steps, what is taking place here. As you all can see here, the megaspore mother cell is always diploid type. They are undergoing with the process of meiosis 1, and they are forming a dyad stage where both the cells, they are carrying n number of chromosomes. After the second meiotic division, we find that all 4 cells have formed, but after this, at the end, we find that only one functional megaspore is present, which is showing maximum growth, but the other 3 cells, they are degenerative type. They are degenerating, but only one is functional here. Now in the next stage, see this functional megaspore, you can see here, that is the first figure. It is undergoing with the process of mitosis, and they are forming 2 cells, 2 nucleus particularly. Again, they undergo mitosis, forming 4 nucleus inside 1 cell, and then 8 nucleated structure. These 8 nucleus which are present here, they get incorporated into certain arrangement. This is the structure of the embryo sac, which you can see here. And as you can see there, 3 nucleus with certain amount of cytoplasmic content, they have come towards the chalazal area, where they are forming 3 antipodal cells. Towards the micropylar region, we find 1 egg cell. This is the most important cell which will be forming the embryo. The 2 synergids are present on the 2 sides, and we find that the polar nuclei right at the center. So here, this is the overall structure of an embryo sac, and this is very important to practice even. The anatropous ovule, inside the embryo, inside the anatropous ovule, this particular structure you need to draw, and you need to finalize that what are the total 8 nucleus present inside them. These megaspores which are present inside the ovary part, that is particularly inside the ovule, as we have seen in this flower. Once again, I am showing you just to have this practical idea that the stigma, the style, and the ovary. Inside the ovary, ovules are present. That means inside these ovules, the embryo sac are present. Now this will ultimately form fruit. The ovary is going to form fruit, and inside the fruit we are going to get seeds. Yes, this is a collection where you can understand that fruits are present. You can see here, this particular plant, they have grown with fruit. So here we can say that megaspore, they are present inside, they are forming the seed. And this is a very important mechanism in case of sexual reproduction in flowering plants, because they will be giving rise to new generations. That means this sexual reproduction will help to maintain the progeny of a plant. So far, we have studied about the different divisions of cell, and we have also seen that how particularly the megaspore cell is getting formed. The overall structure of the embryo sac we have studied. At the chalazal end, we have found that 3 cells are present together and they are forming the antipodal cells, whereas towards the micropylar end, they are forming the egg apparatus, which is consisting of the egg cell and the 2 synagogues. So today we have discussed about the process of megasporogenesis, how megaspore is getting transformed, how the haploid cells are being produced. In this total process of gametogenesis, in the previous class also we have studied about the process of microsporogenesis, where the generation of haploid cell we have seen. Here today also in this class we have seen that how the haploid cells have come up. Now these 2 haploid cells, they undergo with the process of fertilization, and then the next processes of embryo formation starts. But before starting with the process of fertilization, in case of plants, we find there is another very important phenomena, that is pollination. Yes, in our next class, we are going to study about pollination, the different types of pollination, characteristics of different flowers which are having different modes of pollination, so that we are going to study in our next class. Today till this much. If you are getting any queries, please do ask. Till then, bye-bye.